So I'm Jeekin Lau, and I'm a postdoc for Oscar and Dave, and I'm going to be talking a little bit about the genetic structure of roses and genomic selection of some key traits. And just a caveat, we're in the very preliminary stages of using genomic selection, and we're just kind of in the exploratory stage. So this is going to be very preliminary compared to Felipe and Mitchell's presentations. And we're using genomic selection mainly as parental selection. And I have to kind of tell a little story of why we are kind of switching to genomic selection. So this summer, it was a record heat in Texas. And I think we had a record number of days over 100. And so that didn't do very well for a lot of our plants. And a lot of our plants that we had in the field come from biparental mapping populations from me, from other graduate students in the past. And they just didn't look too good. So Dr. Byrne and Oscar, they asked me, they said, hey, we need to rejuvenate these plantings using clonal propagation. And we need to select, we don't have room to propagate all of them and the manpower to propagate all of them. So we need to pick the top 20%. So they said most of these things have been genotyped and phenotyped, and it's just all historical data. It's just sitting there. And why don't you take a crack at it? So that's what I started to do in biparental mapping populations. And we had other populations, a GWAS population that's just historical data just in the past that we had in the program that I was also able to utilize. And I'm going to be talking a little bit about that population, the GWAS population today. So, and of course, every talk that I've been giving at this Tools for Polyploids meeting has been some software comparisons. So of course, there's going to be some software comparisons today in this talk. So a little background about garden roses. Garden roses are economically very important worldwide and in the US, and important breeding goals for breeders are disease resistance, flower productivity, and plant architecture. And talking about plant architecture, if you look at these two pictures right here, this is Dr. Burns standing next to it. This is a 12-foot structure that has a diploid climbing rose growing on it. And this is Dr. Byrne over here in the greenhouse holding a micro mini genotype that will probably get 2 or 3 times that size and is really good for a potted miniature. And so from this picture right here, you just see the genetic diversity that's displayed phenotypically. In addition to these important breeding goals, there's other things like flower color, flower size, flower shape. On this next slide, you'll continue to see more of the phenotypic variation that we have to work with in roses. For example, you can see here that there's plants right here that are completely covered and coral in color. You have red colors, you have yellow colors, you have pink colors, you have white colors. You have plants that are sparsely filled with roses. So there's a lot of variation that we can exploit as plant breeders. Attributing to a lot of this phenotypic variation that we can see is the genetics. So the cultivated garden rose comes from interspecific crosses from 10 different species. So this makes the genetics just really complicated. It also gives us a lot of that phenotypic variability that we see. And rose species are reported from diploid all the way up to decaploid. So there's a lot of different ploidy levels. However, the modern garden rose that you see at Home Depot or at Lowe's, when you go into the garden center, they're mostly diploid, triploid, and tetraploid. So these pictures that you see down here are some of the very first root squashes that I ever did whenever I started at Texas A&M in 2015. And we were doing root squashes to determine ploidy levels so that we were making crosses on the same ploidy level. Of course, now we have other tools, as Chris showed yesterday in Q-Ploidy. And this is the reason why we needed to develop tools like Q-Ploidy, because we have mixed ploidies in rose germplasm. And in addition to that, rose breeders, a lot of them are hobbyists and backyard breeders. And a lot of the times in our program and the hobby breeders, a lot of times they just try the cross. They don't really know the ploidy levels from morphological features of the plant. You can normally tell a diploid apart from the higher ploidy levels, but from triploid and tetraploid, you can't really tell morphologically the differences. So a lot of times the breeders will just make crosses, and if it takes, they go with it. And another interesting thing about roses is that a lot of times triploids can be used for breeding as a pollen parent. So we have all these weird ploidies, and that's why one of the reasons why we shared yesterday, Chris shared yesterday about the Q-Ploidy app. And of course, these pictures right here aren't as beautiful as the ones that they showed yesterday that Oscar did. So I want to give a little bit of introduction about the key traits that I'm going to talk about later for genomic selection. And so the first one is black spotted rose. This is probably the most well-studied fungal pathogen and fungal resistance in the crop. And it is characterized by these black spots. And these black spots really start out really small. So early in the season, the plants will look really great. They won't have anything on them. And after the first rain, a lot of the spores will bounce up from the ground level and infect the leaf tissue. And they'll start out as small lesions. They'll get bigger and bigger. They'll cause this chlorotic response, and they will a lot of times defoliate the plant, especially if a cultivar is very susceptible. And there's been a lot of work done, genetic work done on the resistances from vertical resistance. We have RDR1 through RDR4 already published from different groups around the world. And RDR, I think 5 and 6 are about to come out in publications, not from us, from our collaborators up in Minnesota. Our lab has done a lot of work on horizontal resistances. And so more of a quantitative resistance to this fungal pathogen. And there's been meta-analysis done. So a lot of work has been done in this fungus. On the other hand, you have this other fungal pathogen, Cercospora, which forms these small circular target-like lesions with a necrotic center. And in contrast to black spot, the edges are less feathery and they're more defined circular regions. And very little work has been done on this disease, probably because it doesn't heavily defoliate the plant, and probably just because breeders haven't had a focus on this, because it doesn't, from far away, it doesn't really affect how the plant looks that much. And actually, an interesting thing is there's on plant resistance to this fungus, there's only really 2 papers, and they both come out of our lab, and they came out this year. One on the diploid level by Zena, and one on the tetraploid level by me. Other traits that we look at routinely are defoliation and flower intensity, and these are more complex. These are traits that are a lot of times regulated by many genetic factors, and they can be caused by a lot of different causes. So for instance, defoliation that you can see here, these plants back here that don't have anything, but they look like sticks, it can be caused by biotic causes. So it could be fungal pathogens that cause the defoliation. It can be abiotic causes like heat stress, drought stress, salinity stress, things that go into this heat stress, I guess, complex. And things like flower intensity, the other thing that we want plants that look like this, that are covered in flowers. It also is very complex because it has a genetic component along with biotic and abiotic stressors. So what I mean by genetic component is there are a lot of genotypes that are out on the market that do really well in California in that dry environment. And then you put it in Texas, somehow it looks like these guys over here. It just looks terrible. So there's a genetic component that it has the ability to do really well, to flower really well, but due to other factors, whether it be biotic or abiotic stressors, they don't do well here. So in the past, our lab has done a lot of QTL mapping, and from these QTL mapping studies, we have found a large number of large effect, small effect, medium effect QTL, and so that's why we really want to kind of transition over to genomic selection because we're finding a lot of different QTLs that are varying in their effects. And we want to capture both the large effect and the small effect QTL. So this slide, I know there was a lot of talk about genomic selection, and just for didactic purposes for maybe students here, genomic selection originated in animals, and it actually originated in a study on cattle and milking cattle. So Goddard and Hayes, they were trying to predict the milking ability or the milking breeding value of a bull. And of course, you can't milk a bull. So they used its progeny, its female progeny, to estimate its breeding value. And what you have in genomic selection is a breeding population and a training population. And of course, your breeding population has to be pretty closely related to your training population so that you can make inferences after developing a model. So with your training population, you phenotype it really well. And that's very important that we heard in earlier talks. Really good phenotypes helps you develop a good genomic selection model. And then you have some sort of genotyping. And you combine these two in a linear mixed model to develop a genomic selection model. And what you're able to do is from the breeding population side, you're able to genotype individuals and then using their genotype, predict their performance. And this genomic selection is really good for speeding things up. So for example, earlier I was talking about why we're trying to pivot over to genomic selection. And a lot of the populations that I had made back in 2015, 2016, we're actually just now selecting from those the next round. So it's a 6-7 year breeding process. And like Mitchell was mentioning earlier, we can really speed up genetic gain if we cut the cycles down. So if we can cut that down by using genomic selection and maybe cut it down to 2 years. That would help a lot. So this has been extensively used in livestock and maize breeding and soybean in the private sector. So back when Monsanto was Monsanto, they came up with seed chipping and they were able to already predict the phenotypic performance of the corn before they were even planted the corn. So they would only plant the ones that they knew were going to perform well. This allows us to capture, genomic selection allows us to capture both the large effect and the small effect QTL, which is why we're kind of going towards that. So some materials and methods. We planted back, I think in around 2014 or 2015, a GWAS panel, and it had roughly about 500 individuals, and there were tamu breeding lives, commercial cultivars, and they were phenotyped over the course of 3 years, only during the growing season. So there were a phenotype for every month over the course of 24 months. And we genotyped them using the 68K SNP array and using Q-Ploidy, that software that Chris explained yesterday. We figured out that we had 168 diploids, 54 triploids, and 250 tetraploids. And then from this information, we then called their dosages independently in FitPoly. And then we had to somehow bring the data back together so that we either diploidized everything or we tetraploidized everything. So then After we diploidized everything or tetraploidized everything, we then ran structure, admixture, and principal component on the diploidized data, and just structure and principal components on the tetraploidized data. And so to kind of look at the genetic structure that we had planted here. We then carried out some genomic selection using a GBLUP model implemented in GAPIT, using RR-BLUP, implemented in RR-BLUP, and stage-wise. For genomic prediction, we use fivefold cross-validation for each trait for 1,000 runs. Genomic predictions were then normalized, multi-trait indices were created. And then we did an interesting thing we'll show later on is the genomic predictions were then plotted against the genetic structure analyses to estimate and try to see where our sources of resistances were coming from, and where we were getting a lot of these key traits. So for some just background information, more information about the structure runs, we took a random 2,000 markers out of about 20,000 markers, and we ran a K1 through 10 and 10 reps using parallel structure. And then we used structure harvester to determine how many likely groups we had. And then for admixture, we just ran it as K3, as you'll see in a bit. It was determined by the structure runs. Other than that, to the comparisons here, the 3 genomic selection software that were used, the GAPIT and the RR-BLUP were on the diploidized data. The stage-wise was on the tetraploidized data. So this is the results of the structure analysis, and what you see are 3 main groups, which I alluded to on the slide earlier. And these 3 main groups are in the red is the Rosa rugosa types. The green is the Rosa multiflora, Rosa witcherana types. And the blue are Rosa chinensis and European intergressions. And you see all of these, we had a bunch of admixture too. And so I wanted to plot this against a 2D principal component. And with the 2D principal component, you also see there's 3 vertices of this triangle. There's 3 groups. And highlighted in these different colors are the 3 that I just explained earlier, red as rugosa. And I classify them as belonging to one group if they had about 80% or more membership on those structure plots that you saw earlier. So kind of keep in mind this plot because it'll come into mind. I'll talk about this a little bit later after the genomic selection stuff. Here's just another view. We plotted this against the phylogenetic tree, and then we got our groups. We have the multiflora group, we had the chinensis European group, and the rugosa group here, and a lot of admixture. So pivoting over to genomic selection, we ran a bunch of reliability tests where I masked 20% of the data, and then a random 20%, and then I have on the x-axis the known blues and the GBLUP that was estimated. I ran these all 1,000 times, and this is from GAPIT and GBLUP model, and we're getting about 0.4 for black spot, 0.38 for Cercospora, for defoliation 0.42, and flower intensity 0.46. Now I want you to, as I go from slide to slide, these next 3 slides will look exactly like this, but they're the different methods. And what I want you to see is kind of the cloud of the data and how they're really basically the same for all 3 methods. So this is GBLUP and GAPIT with the diploidized data. This is RR-BLUP. So I'll go back up and back down, up and down. They're basically the same plots. And here we have 0.4 for black spot, 0.38 for Cercospora, 0.43 for defoliation, and 0.46 for flower intensity. Then I reran all the data with the tetraploidized data and stage-wise. And one thing that I have to mention is that for RR-BLUP and for GBLUP models, I had to get the blues from AS Rimmel and then just almost run it as basically like a 1-step model. But for stage-wise, I was able to incorporate it in 2 steps where I include the variance covariance matrices from step 1 into the genomic selection step. And we basically see if I go back up to the R block and to stage-wise, the cloud that we see of data points is the same. And we have 0.41 for black spot, for Cercospora 0.38, for defoliation 0.43, and flower intensity 0.49. Now you'll notice the flower intensity here goes a bit up, and this is better shown in the next slide where I compare the accuracies. So this is each of the traits, and the different colors are the different softwares. And you'll notice here for flower intensity, for some reason, stage-wise, performed a little bit better. And that may just be because it's taking into account the variance covariance matrices and it's using it as a more efficient way to do it. But I'm not quite sure why for that specific trait it has a little bit higher. So with things that are blow up stage-wise, you get marker effects for every single marker. And what I just want to show here on this slide is on the bottom, this is a GWAS using a really basic GLM model. And on the top is the R blood marker effects. And using this slide is just to show that using genomic prediction, we're able to capture the major effect QTL. However, we're able to capture a lot of other things that don't show up, like over here, where there's alleles over here that don't show up here. So there's both positive alleles, there's both negative alleles, and we're capturing both large effect and small effect QTL that may not be detected otherwise using other methods. So after running the genomic selection in all three, so G blip, R blip, and stage-wise, what I did was I got my GEBVs here colored in red. I then normalized the GEBVs and corrected for the directionality. So for black spot Cercospora defoliation, a smaller value is better. For flower intensity, you want a larger value. So after I normalized it, you have to multiply this guy by -1 to get everything on the same direction. And then you can sum them across, or you can apply different weights with the normalized GEBVs, and then you can rank them. So what I did to compare the 3 different, software is I took the top 20% from GAPIT, from RBLUP, from stage-wise, and what we see here is 81% or 85 individuals are the same. This plot over here to the right is a similar plot to, it's just another way to look at this Venn diagram over here. So over here, on this plot on the right, I just arbitrarily picked GAPIT and took its ranking from 0 to 400 something and plotted it on the diagonal. And then I plotted the R, BLUP, and the stage-wise rankings against those rankings. So the genotype is in the same order for all of these, and this is just their rank. What you see here is that GAPIT and, or not GAPIT, GBLUP implemented in GAPIT and RBLUP are essentially the same thing. And stage-wise is slightly different. And this slight difference may be due to it being a tetraploidized data set. And it may be also due to it being a little more, just a different way to run the data. It's a more complex of data set. Also, it can take into account dominance and other gene actions that tetraploids have that aren't accounted for on the diploid models. Now, what was cool about after doing all this, we also had done the structure analysis earlier. So we were kind of thinking, okay, where are some of these resistances, or where are some of these traits coming from? So remember earlier we had mentioned that In this PCA plot, the red was for rugosa, the green was multiflora witchiana, the blue is chinensis, and the gray is admixed individuals. Right here in each one of these plots, I took the top, I highlighted in red the top 20% of individuals that performed for these traits. So for instance, here in black spot, what we see is that the best performing individuals, or the individuals that had the most favorable alleles, came from the rugosa types, and from the multiflora witchiana types, and some from the admixed. Looking at Cercospora, we kind of see it coming from the chinensis and European material. The defoliation, you can kind of see it's kind of equally spread. The red dots are really everywhere. And for flower intensity, we see that it really comes from this this green group, the multiflora witchiana group, and the admixed individuals. And when I make the selection index with all 4 of these traits and pick the top 20%, what happens is you see it's all over the place. So this may be like a good data set to kind of play around with, and I'm going to explore using Jeff's new tool, Coma, to kind of make this more have a more methodical way of selecting individuals to cross. However, like just looking at this, I wouldn't want to keep making crosses in one of the vertices of these triangle, just because you're going to basically bottleneck yourself. So for some summary bullet points is that the diversity set here at Texas A&M is composed of 3 different clusters. There's very little differences that we saw in the diplodized data set and the tetraplodized data sets. And that holds true for looking at the genetic structure and for the genomic selection. And there's very little differences between the results. And we had R squares of between 0.4 to 0.5, some that are close up, and we are beginning to marrying the 2, structure analysis and genomic selection, we're able to start seeing where some of the favorable alleles are coming from for black spot, Cercospora, flower intensity. And kind of what we need to do is explore using Coma to help with the parental selections and what crosses to make to basically make the best genetic gain and to not bottleneck ourselves. So with that, I want to acknowledge everyone in the Rose Lab and the SCRI grants that all went into funding this work, especially Tools for Polyploid and other project members and other collaborators. And with that, I'll take any questions.